So, hello everyone, and happy to see all of you in this new episode of the BioClock Academy lecture series. My name is Juana Georgiana Rus Oswald, and I'm a postdoctoral researcher in the BioClock Consortium. And together with Hannah Brooks, who is a PhD student in the same consortium, we will host the session today. For those who are new, the BioClock Consortium is a Dutch research organization which has the aim to evaluate different aspects of the biological clock in health, in society, and in nature. The aim of our lecture series is to introduce you to the basic concepts and equip you with the proper knowledge from experts in the field. And this lecture series takes place every third Wednesday of the month. And for the time frame today, we will have a 40 minutes talk followed by 15 minutes of discussion, or 10 minutes, let's see. So roughly 1 hour, so that we can close at 5 p.m. Central European time. And during the talk, you can also write your questions in the chat so that we can discuss them later, but also you have the option to unmute yourself at the end. Maybe a good information for you guys, this will be our last session before the summer break, so we're happy that you joined now. And now I will give the floor to Hannah, who will introduce our guest speaker and the exciting topic of today.
Yes, so also warm welcome from me. Today I have the luck to introduce you to Professor Christine Merlin. She's professor at the Texas A&M University in the Department of Biology. And she started her career doing a PhD in insect physiology in France, where she worked on the circadian control of olfactory rhythms in moths. And then after, with her postdoc, she continued to work in chronobiology, but now she started working on the long-distance migration of monarch butterflies and how this is controlled by the circadian clocks. And this is work that she still continues today within her own lab group. So to unravel this quite fascinating phenomenon, I would say, they use a very integrative approach using molecular and genetic approaches, but also behavioral research. And so the main questions that they want to answer is how the circadian clock and the clock genes govern the photoperiodic seasonal migratory switch, and also what the molecular and the genetic basis is for this migratory behavior. And I think, yeah, we will learn all about it today. So I'm very pleased to open the virtual floor to you, Professor Merlin.
All right, thank you so much, Anna and Oana, for inviting me to talk to the BioClock Academy today. And as Anna just mentioned, what I will do today is give you, some flavor of what my lab has been working on. Started from, the very beginning of my career working on monarch long-distance migration and their involvement of circadian clock. And then I'll be moving on to some of the work that we've been doing to understand the link between the circadian clock and photoperiodism, as well as, using the monarch circadian clock as a model system to understand how the clock is ticking, focusing on circadian repression. So I will start by, oh, and I cannot move my slide anymore. So I think I'm gonna have to end show. I don't know why. Stop sharing.
Stop sharing and start again like this.
I will share again. I think this is how we fixed it before.
Yeah, and now?
Okay.
Yes.
All right. So I will start by essentially introducing you to the fascinating long-distance migration of monarch butterfly. So every year, pretty much like clockwork, the monarch butterfly that are present in the northern US undergo this multi-generational long-distance migration, which is triggered by changes in the environment. And it all starts in the fall with decreasing photoperiod, along with other factors, such as decrease in temperature, which really switch the entire behavior and physiology of monarchs that are present at their breeding site in the southern Canada and the northern US. And so in the fall, those butterflies really become very oriented. They present this strong urge to fly south with turning on specific orientation mechanism. They increase their lifespan and they shut down their reproductive status, which we call diapause in insect, in order to allocate more energy towards their demanding flight, allowing them to fly thousands of kilometers southward to reach their overwintering site that are located atop volcanic mountain in central Mexico. They spend the winter at their overwintering site in roost. As you can see here on the picture, those are essentially grapes of monarch butterfly. They are not leaves. And when the spring comes with increasing photoperiod and temperature, they break the diapause, start mating en masse before doing something that is quite remarkable, which is to reverse their flight orientation northward, such that the fertilized female can go deposit their eggs onto emerging milkweed plants, which are emerging at that time of the year in the southern state of the US. And milkweed plants is the only plant onto which monarch larvae actually feed. And from those eggs will eclose a new generation of butterfly that are reproductive. And it takes 2 to 3 generations, as shown by the green and black arrow here, of those reproductively competent monarchs to move northward to repopulate the breeding range in southern Canada and the northern range of the US. And the next fall, when a proper environmental condition occurs again, a new migratory cycle can start. And for a long time, we've known that the circadian clock is actually playing a central role in the ability of monarch butterflies to navigate in an oriented fashion in the fall, as well as in the spring. And we know that monarch actually use the sun as the primary compass cue to navigate. But because the sun is a moving target across the sky over the course of the day, they have to time compensate for the azimuthal position of the sun changes over the course of the day. And to do so, they use the circadian clock. And this was originally demonstrated very nicely by flying fall migrants in this Moritz and Frost flight simulator here under natural sky. And so, as you can see, this is a very simple large plastic barrel in which a butterfly is essentially suspended by a tether that is glued to its thorax and allowed to fly in all positions on the horizontal plane. And this tether is essentially hooked up to an encoder that allows the experimenter to record flight orientation that a butterfly is taken relative to a starting position. Okay. And essentially using this behavioral apparatus, what's been shown is that the clock is absolutely required for this behavior. And I'll show you in a cartoonish way how this was demonstrated. So if you capture fall migrants and bring them in the laboratory and train them to LD cycle that are just in sync with the prevailing LD cycle that I encounter in the wild, those butterflies, those fall migrants, orient themselves when flown in the afternoon and the sun is in the west, fly towards the south-southwest. Now, the role of the clock was initially shown by actually doing an experiment that is very prevalent in our field, which is to do a clock shift. And so if you now capture this butterfly, bring them in laboratory condition, but then train them in an incubator where the LD cycle is now delayed by 6 hours. That is, the light in the incubator will come on 6 hours later than it does in the wild. These butterflies shift their flight orientation clockwise by about 90 degrees. So they shift by about 15 degrees an hour, moving towards the north-northwest. In essence, shifting their flight orientation such that the sun, that was on their left shoulder, is now on their right shoulder. So they interpret this afternoon sun as being a morning sun because their endogenous clock is telling them that this is morning time. And the role of the clock was nailed down by essentially subjecting monarch to constant light, which is a way to kill the functioning of the clock. And under those conditions, fall migrants were now no longer able to time compensate and simply follow it followed the sun, irrespective of the day at which they were flown. We also know that monarch butterflies do so with a very unique clock, in that the component of the clock, the makeup of their clock, is actually a stripped down version of the mammalian circadian clock, where mammalian-like components are actually conserved. So if you look at the relatively simple transcriptional translational feedback loop that forms the clock in mammal, you see that activation is provided by the clock BMAL1 heterodimer, which binds to E-boxes in the E-box element in promoter region of repressors that essentially activate their rhythmic transcription in a rhythmic fashion. Those repressors are several period genes and several cryptochrome genes. Upon translation, they form these complexes in the cytoplasm and shuttle back into the nucleus to go inhibit their own transcription and the transcription of other clock control genes in about 24 hours. And what's very special about the monarch clock is they navigate with a mammalian-like clock in that they have retained the mammalian-like component, including BMAL1, whose C-terminal transactivation domain had been lost in other insects such as Drosophila. And they also retain the mammalian-like circadian repressor cryptochrome that is called cryptochrome 2 in the monarch. And that has also been lost in Drosophila. But what you can see here is that as opposed to mammals, which have several copies of each of the circadian repressor period and cryptochrome, the monarch has a single copy of each, a single period and a single mammalian-like repressive cryptochrome, making it a very interesting model to understand circadian repression that is relevant to humans. So in the first part of my talk, I will go back then when I actually was starting my career working on monarch butterfly as a postdoctoral fellow. And the first part of my talk will focus on the work I've done trying to understand more about circadian clock in time compensation of sun compass orientation. And this all came from the fact that, as Anna mentioned in her introduction, I came from a background working on circadian control of olfactory rhythm in moth as a PhD student. And when I joined the lab of Stephen Rippert, we found in the book written by the great grandfather of monarch biology, Fred Urquhart, some anecdotal observation that he made more than 70 years ago now. And what we found was this quote in the book that said the antennae may play a role in the navigational abilities of migratory monarch butterflies. And with my background on antennal clocks, I really wanted to test this hypothesis in control condition. So to do this, we flew intact butterflies as well as butterfly from which we clipped off the antennae in the Moritzen Frost flight simulator under natural sky, as I showed you before. And there are two paradigms, a paradigm where the butterflies were entrained under prevailing fall-like LD cycle, and a paradigm in which we clock shifted the butterflies by a 6-hour delayed LD cycle. And so, as is typical for intact fall migratory butterflies, we find that under prevailing LD cycle, so fall-like LD cycle, these butterflies were oriented significantly as a group towards the south-southwest. The individual dots that you're seeing on the perimeter of the big circle represent individual flight orientation, and the arrow represent the mean orientation of the group, with the length of the arrow representing the significance of this orientation. And as expected, under clock shift by a 6-hour delay, these butterflies were shifting their flight orientation clockwise. Here, overshooting a little bit from 19 degrees, but you see that these butterflies were also oriented as a group. So these butterflies were able to time compensate their sun compass orientation. But surprisingly, when we flew antennaless butterflies, which was done, intermingled with these intact ones, we find that antennaless fall migrant were completely disoriented as a group. As you can see, all those red dots here, individual flight orientation, were pretty random, irrespective of the LD cycle in which they were flown, although every single individuals here were flying as strongly and as in an oriented fashion as their intact counterparts, suggesting that the antennae were indeed necessary for sun compass orientation. So one thing that we wondered first was whether clipping off the antennae could impede the proper functioning of the brain clocks, because at the time, the only clocks that were not control behavior were known to be located in the brain. So to test this idea, we essentially quantified the expression level of two core clock gene, period and timeless, in the brain of both intact and antennaless butterflies. And as you can see here, under LD condition, right? And as you can see here, is that both per and timeless were robustly cycling in the brain of both intact and antennaless butterfly, suggesting that the brain clock were functioning normally in antennaless butterflies. So the next possible hypothesis was perhaps this antennae actually harbor circadian clocks that could function independently of the brain. And I'm not showing you the data. This is work that is, that has been published more than 15 years ago now. But we've shown that Monarch indeed possess antennal clock that function independently of the brain clocks by essentially culturing antennae, in dishes and showing that they could retain for many days the expression of rhythmic expression of core clock components. So this really led us to ask the question, are antennal clocks involved in time-compensated sun compass orientation? And of course, at the time we were working in a species in which genetic tools were not available. And so we had to be essentially crafty and use the tools that we had, in our toolkit. And this was paid because one property of circadian clocks is that those clocks are entrained to light during the LD cycle, which generate rhythm. But if after entrainment, if the individual are maintained, or those clocks are maintained under constant darkness, the rhythmicity actually persists for many days. So what we decided to do was to actually paint the antennae black in order to block the light input to the antennae, in order to alter the rhythmicity of antennal clock, and use a clear paint as a control. We first tested whether this treatment would essentially affect the functioning of the clock as we expected it should. And indeed, we quantified, again, per and timeless mRNA level expression over the course of 24 hours in both clear painted antennae and black painted antennae 11 days after painting. And what you can see is that both per and timeless actually were expressed rhythmically in a robust fashion, but in black painted antennae after 11 days, this rhythm was severely dampened, either due to individual clocks started to get out of sync with one another, but you see here non-significant but residual rhythm, right? So the ability to desynchronize circadian clock in the antennae allowed us to test whether the synchronization of antennal clock would alter flight orientation of all migrants. And so what we did was to fly these clear painted antennae butterflies along with the black painted antennae butterflies again under fall-like LD cycle or clock shifted fall-like LD cycle. And we use non-painted butterflies as a control. As you can see, those non-painted butterflies were again oriented significantly as a group, although those oriented towards the south-southeast in this particular experiment because they were flown later in the season. And we know that late in the season, this is what we usually see in terms of shift of flight orientation, although we don't know the cause of that. In any case, when we flew clear-painted and teeny butterfly, just as not painted clock control, again, under 4-like LD cycle, those guys were oriented towards the south-southeast and were able to clock shift by shifting their orientation by about 90 degrees clockwise under 6-hour delays 4-like LD cycle. Now, to our surprise, when we flew black-painted and teeny butterflies, we find something unexpected, which was that the black-painted butterflies were actually flown under 4-like LD cycle with lights coming on at 6 a.m. in the morning, were actually oriented as a group, but were shifted their orientation about 180 degrees compared to the clear-painted butterflies, and under clock shifted condition were actually reversing their flight orientation back to the southwest. And interpreting this data were a little puzzling at the time, but we soon realized that those butterflies, those black-painted butterflies, have actually been flown over a pretty substantial amount of days and their clocks were actually free running in constant condition. So one hypothesis that we tested was whether this altered flight orientation could be due to free running internal clocks. In other words, would there be a correlation between the direction of oriented flight and the day of study after painting? So to test this idea, we performed essentially a correlation between the orientation taken between butterflies and the day of study after painted, but we analyzed the data in a counterclockwise fashion, as we know that the free running period in monarch butterflies based on eclosion behavior data is actually a short period. So every day the period should get shorter and shorter. So this correlation should be done in a counterclockwise drift over time. And when we did that, we indeed find a correlation suggesting that the internal clock were actually providing the primary timing mechanism to this sun compass system for oriented flight. And so here is the summary slide that shows our understanding at the moment of the circuitry in the brain that is involved in skylight cues integration, where sun compass information is actually integrated, and the knowledge that this paper provided strongly suggesting that internal clock in the antennae were the primary clock providing timing information to the sun compass system to essentially have butterflies oriented in particular orientation. The way by which these internal clocks are actually communicating, the neural basis of this communication to the central complex remain unknown at this time, because we do not have the proper genetic tools to actually mark those clock neurons in the antennae and trace their projection in the brain. And so this was a part of my postdoc, and I spent many years after that, still as a postdoc, to essentially try to develop the monarch butterfly along with colleagues in the Stephen Rippert lab as a model system, as a genetically tractable model system. So what we did was sequence the monarch genome. We have now a chromosomal level assembly of the monarch genome. And I spent many years developing reverse genetic tools using all possible generation of engineered nucleases to be able to generate knockouts from laboratory-raised colony, which I also had to develop. And this was really a stepping stone to set up my own lab using these reverse genetic approaches to try to understand how the clock is involved in many different aspects of monarch migration, as well as how circadian repression is achieved at a mechanistic level. So in the second part of my talk, I will illustrate how we leverage those genetic tools to actually try to link circadian clock to the season. And the work that I will show you is work that was done by a very talented former grad student in the lab, Sam Mayams, who's now doing our postdoctoral studies in the lab of Joe Takahashi. And what Sam spent our PhD doing was essentially testing the idea that circadian clocks may be able to sense changes in photoperiod between long and short photoperiod, which should lead to these reproductive diapause that we found actually in fall migrants that are subjected to short photoperiod. And if circadian clocks in the brain were actually necessary to sense difference in photoperiod, which was not known at the time in any insect species, she hypothesized that perhaps this clock would drive the expression of clock control genes that would be regulated in a seasonal fashion to give rise to a reproductive status under long photoperiod or a reproductive diapause under short photoperiod. So to test this, the role of the circadian clocks in photoperiodic responses, she used 3 different knockouts that we had previously generated in the lab. 2 mutants that essentially abrogate activation of the clock, a clock mutant, a clock knockout, as well as a BMAL1 C-terminal truncated mutant that lacks the C-terminal domain that is known to be necessary for the clock BMAL1 transactivation. And she also used a Cry2 knockout, the knockout of the major repressor of the clock, as I demonstrated during my postdoc. And raised both female of all of these knockouts along of their wild-type siblings under either long photoperiod with 15 hours of light or short photoperiod with 10 hours of light. And as you can see, and expected based on the literature, she found that wild-type female raised in short photoperiod were developing significantly less mature oocyte than female raised in long photoperiod. But interestingly, the Cry2 homozygous female, unlike wild-type siblings, were losing their ability to sense the photoperiod and were producing even less mature oocyte than the female raised, the wild-type female raised in short photoperiod, suggesting that Cry2 knockout may be locked in a diaposing-like phenotype. When she tested mutant for the activators of the clock, and this BMAL1 C-terminal truncated mutant, she found that just as Cry2, those were also losing their ability to discriminate between long and short photoperiod, but were producing as many mature oocyte as the female raised in long photoperiod. And because the phenotype of this knockout for activators and repressors, which have opposite function in the clock, are also going in opposite direction, we think that this was strong evidence that the clock as a functional unit was involved in sensing difference in photoperiod. So Sam went on to try to identify genes that would be under seasonal regulation using RNA-seq at a fine temporal resolution from the brain of different group. So she did RNA-seq every 3 hours over the 24-hour LD cycle from the brain of monarch that we raised in the laboratory condition under LP, so long photoperiod or short photoperiod condition at constant temperature, and summer and migrant butterflies that were caught in the wild. She, of course, started by verifying that both these groups were presenting this nice expected reproductive diapause under short photoperiod, and went on to do libraries, sequencing, and performed an analysis of rhythmic gene expression. And what this revealed was a molecular signature of seasonally regulated gene expression. So what you're looking at here are heat map of gene expression in each of the conditions with 2 biological replicate plotted next to one another. Each line on the x-axis, on the y-axis, represents the expression of a single gene for a single replicate 8 time point over the first 24-hour cycle, LD cycle. And high expression is represented in yellow, low expression of the gene is represented in blue, and those gene expression, each line represents the expression of a different gene, And from on the y-axis, this is arranged by peak of expression. So gene peaking in their expression early during the day should be found at the beginning of this plot. And so in essence, what you're seeing when you're looking at yellow lines interspaced by blue lines, you're looking at genes that are rhythmically expressed, okay? So what Sam found was essentially 3 categories of genes from both monarch raised in laboratory condition or monarch caught in the wild. The first category were genes that were rhythmically expressed in both conditions. The second one was genes that were rhythmically expressed in the brain of monarch raised in long photoperiod or in summer butterflies, but whose rhythmic expression was severely dampened, and we call them arrhythmic here for simplicity, in the brain of monarch raised in short photoperiod of migrant. And the third category of genes were genes that were rhythmically expressed in the brain of monarch raised in short photoperiod and migrant, but not for those raised in long photoperiod or found in the summer. And the reason why we essentially did those 2 individual data sets was for us to be able to overlap them in order to be able to pull signal from the noise, hypothesizing that if the clock truly controls the seasonal regulation of gene expression, it should do so in the same way from both monarch that we raise in the lab and monarch that we capture in the wild. And the category that we were really interested in was this rhythmic arrhythmic category, because work in mammals had shown that genes that are under photoperiodic control are usually rhythmic in long photoperiod, but their rhythmicity is dampened in short photoperiod. So Sam looked at this category and overlapped the 2 data sets, which, as expected, significantly reduced the number of genes that would be found in this category. She found 33 genes that were rhythmic in all seasonal form, 22 that were only rhythmic in the brain of monarch raised in long photoperiod or in summer, and 12 that were only rhythmic in the brain of monarch raised in short photoperiod or in migrant. But as I mentioned, the category of interest to us was this rhythmic arrhythmic category. Of course, this gene list is too small to actually be able to perform enrichment analysis. But simply looking at the list, we find that 3 genes out of the 22 were actually belonging to the same pathway, which is the vitamin A pathway. Santa Maria 1 and 2, as well as retinol dehydrogenase 13. And interestingly, the rate-limiting enzyme of this pathway, LINA-B1, was found to be expressed rhythmically in all seasonal form. So what is the vitamin A pathway in insect and where it's found, how it's working? So essentially, it's found in extraretinal cells in the head. Santa Maria encodes a transmembrane transporter for beta-carotene, which allows the uptake of beta-carotene in the brain. Once uptaken, this beta-carotene is converted into retinol by the rate-limiting enzyme LINA-B. And in insect, we know that retinol can be interconverted by retinol dehydrogenase into retinol. Now, we also know, based on work in seasonal mammal, that retinol can be converted into retinoic acid by the retinol dehydrogenase enzyme. And this retinoic acid is a very interesting molecule, regulated, seasonally produced in mammal, that can go on to bind to retinoic acid receptor that are transcription factor that regulate downstream transcriptional program. Okay, so of course, Sam had demonstrated that the vitamin A pathway was under seasonal regulation. She also wanted to test whether it was under circadian control. And to do that, she leveraged the availability of another RNA-seq data set that we've generated in the lab from brains of monarch knockout for Cry2 and clock. So both the repressor and activators of the clock. And you can see that all of these genes show a nice rhythmic expression in the brain of wild-type butterflies, but their rhythmicity is completely abolished in the brain of both clock knockout and Cry2 knockout, showing that they are not only under seasonal regulation, but also under clock control in the brain. So all of that was really interesting. But what we wanted to demonstrate next was the effect of genetic disruption of the vitamin A pathway to test whether it would be really necessary for photoperiodic responses. So to do that, Sam generated a CRISPR-Cas9 mediated knockout of the rate-limiting enzyme NINA-B and raised those females in long and short photoperiod along with their wild-type sibling. If retinol and the seasonal regulation of retinol is necessary for photoperiodic responses, then those knockouts for NINA-B should lose their ability to distinguish between long and short photoperiod. And this is exactly what she found with NINA-B female producing in both photoperiodic condition as many mature oocytes as their wild-type sibling raised under long photoperiod. So just to summarize this part, we found that we demonstrated that the circadian clock is involved in sensing photoperiodic changes in the monarch, that this clock regulates the vitamin A pathway, which is itself seasonally regulated, and that this seasonal regulation is necessary for photoperiodic responses. For the sake of time, I didn't show you the data, but we've also eliminated the possibility that this response could be mediated via the compound I, so we know that this is happening really from the brain. And my lab is currently following up on this work, testing the non-mutually exclusive hypothesis that the vitamin A pathway could function either via the seasonal production of a deep brain photoreceptor, retinol being the chromophore of these receptors, or via the regulation of gene expression by retinoic acid, which could then lead to this downstream seasonal regulation of gene expression in the brain. So how am I doing in time?
I'm
doing
40 past now. So still about 10 minutes left.
okay. So
10 minutes questions.
All right. So in the third part, I just want to shift gear here and really talk about some of the work that we've done to use the Monarch circadian clock to study circadian repression, which is not fully understood at this point, especially in mammals, because of the complexity, right, of the circadian repressive complex with many different copies of period and cryptochrome that all have similar but different function, right? There is redundancy with slight differences between these different paralog. And so what my lab has been doing and the work I'm going to talk about has been led for many years by a senior research associate in the lab, Ying Zheng, is done in collaboration with our renowned colleague who's working in Drosophila and who was responsible for the discovery of the circadian feedback loop originally in Drosophila, Paul Hardy. So my lab has been focusing essentially on two main questions. How is cryptochrome 2 and period repression of clock and BMAL1 achieved at a mechanistic level? I'm not going to spend time talking about cryptochrome 2 because this has been published many years ago. The only thing that I want to emphasize here is that using the Monarch as a model system allowed us to validate findings that were found in vitro in mammals, but in vivo for the first time. And what we've shown is that this mammalian-like cryptochrome actually repress and regulate the circadian period of the clock by binding to the BMAL1 C-terminal tail that was shown in mammal to actually be the site of repression, but we shown in vivo generating mutants of this tail domain that this is indeed where cryptochrome actually binds to initiate cryptochrome repression. Now, what I want to spend time on is what we've done more recently in collaboration with Paul, to try to understand how period repression of clock and BMAL1 is achieved at a mechanistic level, because despite being one of the first clock gene to have been ever cloned and leading to the concept of this 24-hour feedback loop, we still know very little about how period is actually working to achieve repression. And so we started working on that with the idea that perhaps per could repress or per repression could occur through the other non-activation domain that is present on clock in BMAL1. I just told you we know that cryptochrome actually repressed by binding the C-terminal domain of BMAL. But we know that there is also an activation domain on clock known as the clock exon 19. And we thought that perhaps those two different repressors would have converged function onto two different activation domains of the clock BMAL1 heterodimer. So we know based on work in mice generated here from the lab of Takahashi that mouse that are mutant that lack the exon 19 actually shows this long period phenotype and eventually become arrhythmic if you let them free run for long enough. Work from the late Paolo Sassone-Corsi's lab also showed that this clock exon 19 was necessary for the establishment of a permissive chromatin state for circadian repression via the binding of the histone methyltransferase MML1 to this domain. So when MML1 binds to clock via the exon 19, you have nice circadian transcription. When it does not, circadian transcription is severely dampened. The question that we wanted to started asking essentially was, could circadian activation and repression by pair also occur through the exon 19 in the monarchs? And it turns out, as you can see here on the alignment, that this clock exon 19 domain is actually very well conserved between mammalian clock, monarch, shown as DP clock here, and Drosophila clock. So Ying started by using the CRISPR-Cas9 system to generate a monarch mutant that would lack the exon 19 analogous region of the mouse clock exon 19. And you can see here the mutation that she recovered that is essentially reminiscent of the mutation that had been generated for the mouse delta 19 mutant. And she tested this monarch mutant for defect in circadian period by essentially looking at the eclosion behavior of monarch, which we know is under circadian control, under constant darkness, at both DD1 in circadian in constant darkness after entrainment, or DD3 to be able to derive change in circadian period. And you can see in black here that wild-type butterflies, as expected, were showing a nice peak of eclosion. And I must mention that here this assay is a behavioral level, is a population level assay. You're looking at a population of eclosion, not individual assays such as locomotor activity rhythm. But what you can see is at both DD1 and DD3, the time at which these butterflies were eclosing was not different. However, started looking into heterozygote mutant in gray or homozygous knockout in red, you can see an increase, right, of the circadian period. So lengthening of the circadian period in this monarch delta 19 mutant, a phenotype that phenocopies the mouse clock delta 19. We also showed, and I'm not going to just go into detail, that the clock in this delta 19 mutant was also abolished in the brain by quantifying timeless expression and other clock gene expression. What we went on doing was to then turn to in vitro assay to actually test our hypothesis that maybe the clock exon 19 analogous region could be necessary for power repression. So to do this, what we do is co-transfect Drosophila S2 cells with monarch clock component, along with a luciferase reporter that is driven by e-boxes onto which the clock and bmol1 is actually binding. Okay, so open binding of clock and bmol1 to this luciferase reporter, you see nice activation, and open co-transfection of increasing doses of PER, you see this dose-dependent repression. Okay, now when we transfected S2 cells with a clock that was missing the exon 19 analogous region along with BMO, we saw, just as shown in mouse, a severe decrease in activation. But very interestingly, PER repression was completely abolished, even when we tried to repress with high doses of PER. And this was specific to PER, because repressing with increasing doses of the other circadian repressor Cry2 was capable of nicely repressing this clock delta 19 mutant BMAL1 heterodimer. And it can do so because Cry2, as I mentioned before, is actually repressing on the BMAL1 molecule. We went on to test whether trithorax was also necessary for PER repression. I told you trithorax is this histone methyltransferase that binds to the exon 19 for activation. And again, we tested that in Drosophila S2 cells. And we tested that by knocking down endogenous trithorax. As you can see here in red, when we knock down endogenous trithorax, activation of clock and bmol1 is again severely decreased and repression is completely abolished. So knocking down trithorax has the same effect as removing clock exon 19. Now, as you can see here on the cartoon on the left, this trithorax is actually an enzyme that has a catalytic activity, okay, that is present on the C-terminal domain. And I don't want to enter too much into detail here, but in human, a single residue on this catalytic active domain, known as the Z domain, had been identified. And a single point alanine mutation was sufficient to abrogate catalytic activity. This residue in Drosophila trithorax is actually conserved. And so what we actually did here was to try to see whether we could rescue the defect in activation and repression when we knock down endogenous trithorax by adding back either a wild-type trithorax or a catalytically inactive trithorax. I'm not showing you the N-terminus here. We only use the C-terminus. You can see, focus on the gray graph. When we add back a wild-type C-terminal domain, we indeed rescue both activation and repression by PER. But when we add a catalytically inactive trithorax, where we mutated this residue at position 3665 to an alanine residue that cannot be methylated, we to some extent rescued activation, but not PER-dependent repression. We also showed that this catalytic activity is required for PER-clock interaction by performing co-immunoprecipitation of PER and clock in absence or presence of this RNAi knockdown of endogenous trithorax with the rescue with a wild-type C-terminal domain or a catalytically dead one. We probe the interaction using an antibody against PER and perform Western blot to look whether clock was actually binding to PER in our pull down. You can see that in presence of endogenous trithorax, when we pull down PER, clock is binding to it. This interaction is actually gone. You see no clock signal here. When we knock down trithorax, we can rescue this interaction when we add back a wild-type catalytically active C-terminal domain, but not when we add back a catalytically inactive C-terminal domain of trithorax, really showing that the trithorax catalytic activity was required for PER-clock interaction. And this really led us to wonder whether trithorax could methylate a non-interactor that would be necessary for clock per binding as well as per repression. So to test this, we repeated this co-immunoprecipitation experiment. But in addition to that, we decided to probe the pull down with an anti-methylated antibody against lysine residues. And the reason to go after lysine residues was that trithorax is an histone methyltransferase that is known to methylate on histone on lysine residue. And the expectation was that if the catalytic activity of trithorax is actually indeed methylating a non-interactor, we should find whenever we have interaction between clock and per, right, here and there, we should find proteins that are methylated, but they should not be present in absence of clock and per binding if they are really required for this binding. So we probed the pull down with this anti-methylated lysine antibody. And as you can see here, there is absolutely no difference, right? Our expectations were not met by this experiment. And so we scratched our head for quite some time on that. But it turns out that there is another residue that can be methylated, which is an arginine residue. So we decided to probe again this co-immunoprecipitation now with an anti-methylated arginine antibody. And just as we expected, this allowed us to identify essentially protein that were methylated in presence of the catalytic activity, the catalytically active trithorax when clock and per are essentially binding. So because I think I'm running a little late, I'm not going to go into great detail of how we identified those proteins. We performed mass spectrometry looking for proteins that were methylated and focused essentially on this HOT protein 68. When we performed, again, in vitro assay to look for effect on activation and repression of this HSP68 that was methylated on a single residue, a single arginine residue at position 45, we found that, again, knocking it down was just phenocopying what we find when we remove clock exon 19, when we knock down endogenous trithorax, in that activation was again severely decreased and repression was completely gone. When we add back a wild-type HSP68, we could rescue activation and repression. But when we essentially remove the methylation of this HSP68 at the single residue that was methylated, position 45, by mutating this arginine into an alanine, we could rescue activation, but per repression was completely abolished. And so this really led us to this model of HSP68 dependent per repression, where we think that upon binding of this histone methyltransferase trithorax to clock exon 19, you have nice circadian activation, just like it is the case in mammals. But upon binding to clock, the catalytic activity of trithorax would then either methylate HSP68 on arginine 45, either directly or indirectly through the recruitment of protein arginine methyltransferase, which we are currently testing that. And this methylation of HSP would allow its binding to the complex, recruitment of per, allowing per to exert its function as a circadian repressor. And because, of course, HSPs are chaperoning molecule, and per is a largely intrinsically disordered protein, we are currently working on the hypothesis that maybe the role of HSP in this complex is to actually lead to conformational changes of per that would allow him to work and interact with other proteins in the complex to affect repression. So I will stop there by acknowledging folks in my lab who contributed to the work I presented, current lab member, past lab member, a lot of collaborators, they are not all listed here, as well as the funders who are actually paying the bills and allowing us to do the science we are doing. So thank you all for your attention, and I'll be happy to take any question and stay longer to do so.